from the UK originally. She moved to Canada back in 2001, is that right, to University of Calgary, and then started up at University of Guelph in 2007 while I was a graduate student there. And so she's done a lot of interesting work in the gut microbiome area, and particularly in the area of doing in vitro growth experiments with these bacteria, trying to simulate the gut environment and use that as a technique for studying all kinds of interesting interactions that we and so with that,
okay, thank you very much. All right, thanks everyone, thanks for coming, and I hope you can all hear me okay. I'm gonna just apologize in advance. I'm getting over a cold and it's come with a really nasty cough. So if I start coughing in the middle, I'm very sorry. I am fine. I'll just need to have some water and some cough lozenges. I will be okay, but I apologize because it's something I can't control and it's very annoying. And I'm sure everyone knows how I feel. So there we go. Anyway, so it's been a pleasure to come here today. I had no idea that you guys even had your own airport. It's really kind of fun to find. So it's kind of interesting that it takes 5 hours roughly, so Google says, to drive from Guelph to Penn State. It probably takes about 7 hours all said and done to actually take the flights and queue up at the airport and everything to do that. But I chose to fly for whatever reason, and it's been a pleasure, and the weather's been great. So apart from maybe a bit of rain today, that's fine. So thank you for having me. So I'm going to talk a little bit, as Daryl says, about some of the work we're doing to grow microbial communities in vitro, to do some modeling. But I'm going to give it a very sort of high level view, and I'm going to tell a couple of stories from the lab, some of the research that we've been doing where we've got some results. There's a lot of stuff that I'd love to tell you about, but that I can't tell you about, the reason being that in 2013 I spun a company out of some of the research we're doing. And the company is called Microbiota. It's great because it's doing really rather well, but a lot of the work that we're doing is still a little bit shrouded in secrecy. So I have to kind of take that aside and just tell you about what I'm doing in the academic world. But I'm happy to answer questions as much as I can when it comes to it at the end. All right, so I'm talking to a microbiome group, so I really don't have to show you this slide, but it is important to just sort of bring it up front to have everyone recognize that we are superorganisms of human and microbial cells. In other words, we're not just human. We actually just are collections of cells, really, and those cells aren't just human cells. We exist in a very delicate equilibrium between the host and microbial cells. And most of the microbes that we have on and in our bodies actually reside in our gut. The most diversity is found in the gut, and that's kind of what led me to working in the gut microbiome field back in the time when it wasn't very sexy to do so. So I was really lucky that I kind of got in on the ground floor there, and now it's obviously a very exciting area of research. So every gram of feces contains around 10 to 11 bacterial cells. It's a very high number. And that's just the bacteria. You mustn't forget there's other things in there as well. There's some archaea, there's some yeast, there's lots of viruses. And we're starting to work on all of those things in my lab, but I'm not going to show too much data about that today. I want to focus on the bacteria just from the point of view is that they're the most sort of tractable group to do experiments with, and that's kind of the stories that I'm going to show. I think in some early literature, there were reports that there were about 1,000 different species in the average person's gut. That has sort of been debunked recently. It's more like about 200 species. The earlier estimations are probably due to sequence error from some of the earlier next generation sequence methods that were used. So 200 is about, roughly what the average person has. But we shouldn't sort of say that that's somehow diminished because in fact, for every species you probably have multiple strains, and we don't actually know very much at all about that strain diversity within these different species. And that's something that we've just started to tackle in my lab and has become suddenly very important. And there's some interesting new papers have come out, not of my lab, of other people's labs showing similar things as well. So I think I should watch that space, and that strain diversity is going to become something very important soon. Now, everyone's gut composition is different. I'm sure that if you're in the microbiome group, you've seen this slide somewhere along the line. This is some data from the Human Microbiome Project paper that was published in 2012 that I was part of. And I didn't do the sequencing part of it, but this is the result of the sequencing, this metagenomic sequencing that was done of the gut components of the 139 individuals who were sampled as part of this project. And what you see here is a bar plot. And on the bar plot, you can just see that with the 139 people going along the x-axis, you can see that everyone is different. The point of this isn't to show you that everyone's gut composition is different. It's really to show you this, which is because this was metagenomic data, you could then take the genes and predict their function. And when you do that, you can see that there's an awful lot of functional redundancy within the ecosystem. And if you look at the functions, you can see that even amongst these very different individuals, very different microbiomes, their microbial function is fairly similar. And I think that that's something that you should probably remember going forward in this talk too. Now, excuse me, ecosystem biodiversity drives the overall health of an ecosystem. It's been known for a while. I mean, it's a very kind of generic statement, and I'm sure ecologists among you will probably argue with me for that. But certainly in the gut, it does appear that a high diversity of species drives a healthy ecosystem. It provides balance and functional redundancy that in turn is related to a high gene count, and in turn that provides resistance to damage. And if you contrast that to an ecosystem that has a low diversity of species, you can see that the opposite is true. These ecosystems are generally fairly sick. They are in imbalance. They're usually sort of up one day, down the other day, and the blooms and things are sort of happening very quickly and without any kind of pattern to them. There's functional disability, which comes with a low gene count, and there's susceptibility to damage. So now if you lose a major component of that ecosystem, you could get an ecosystem collapse much more easily than you could if it was a really rich, diverse ecosystem. This is also true, you know, there's pictures of a rainforest and a kind of an apocalyptic landscape there, but this is also true at the microbial scale. So these, the ecological forces that drive this are true at both scales. One of the big questions in the human microbiome field right now is are we damaging our health by eroding microbiome diversity? So this kind of dates back to, most people date it back to something called the hygiene hypothesis that was brought about by Strachan in 1989, so it wasn't that long ago. The thing about the hygiene hypothesis is it actually doesn't have anything to do with the microbiome if you really read about it. What it says is that if you have a lack of exposure to certain infectious agents during your childhood, that is going to drive allergic disease. That was all that was said, and that was what kind of started the whole thing. Until Rook in 2003 developed something that he called the old friends hypothesis. And this is stating this is the first time that the microbiome really kind of hit this field in a way. If you think about it, that happened in 2003. That wasn't that long ago. This is really talking about, for the first time, the idea that humans are dependent on a co-evolved microbiome to educate the immune system and to prevent inflammatory disease. So this is talking more about microbes, less as pathogens and more about old friends. And then finally, we have the missing microbiota hypothesis. This is from Blazer and Falco in 2009. And this is a little bit more of a frightening thing from the point of view that we have they're saying that we have a loss of microbiota that has happened over generations now. And recent changes in our lifestyle have, like taking drugs or sitting watching movies and eating hamburgers all day, have greatly exacerbated this loss. And so what's frightening about this hypothesis is it's talking more about having a loss that kind of compounds over generations. So that one generation may have a poor or a good microbiome, but then was perhaps treated with antibiotics or whatever. And then that lack of diversity as a result of that treatment was then passed down to the next generation and so on and so forth. And so we're seeing a compounded effect. And that really is truly a frightening thing. And one of the things that has come from that is the idea that some of the chronic diseases that we're now seeing in society, like allergy, asthma, that are all rising in their incidence, could be to do with the fact that our microbiome, as a consequence of a much cleaner lifestyle that we lead these days, actually driving driving that process. And so you can conceptualize this. This is actually a figure that my postdoc did for a review article not too long ago, but it's based on David Relman's idea that microbes exist in communities when they are, you can draw them like as these ball and cup diagrams. So there at the top left, you can see that we have what he's tried to depict there as a very complex community. And that sits inside a ball, which sits inside a cup, which is very deep. And that just means that it becomes very difficult to push that ball out of the cup, okay? Now, each time that you remove some of the diversity from the ecosystem, you make the cup a little bit more shallow. And so you make it so shallow that it reaches a point that just a little bit of stress can push it out of the cup into a different ecological state. And so you drive this sort of stepwise evolution of a microbiome being destroyed to the point where the likelihood of disease becomes a lot greater, because the less diverse an ecosystem is, the less stable it is, the more likely it is to be damaged by perturbation or stress. And there's lots of things that can add to that, or that can contribute to that kind of stress. Things like lifestyle, we've talked about some of the things there, but the use of medication, and that includes antibiotics. But there are lots of medications now that we know actually affect the microbiome, which have never been tested for their effects on the microbiome, that we should perhaps be a little bit more concerned about. Diet is a major moderator. I don't think we have to be blind to realize that in the Western world we have a pretty terrible diet. I think that could be improved. We should be eating a lot more fiber than we do. Host genetics plays a role, health and disease. So if you have some underlying comorbidity, that can affect your microbiome. And of course, your age. As you age, your immune system goes into immunosenescence, and the ecosystem changes again the older that you get. And also at the other extreme, in young life, when you're first born, that developing microbiome is quite different. So there are several examples of diseases that are associated with altered gut microbiota diversity. And a lot of these actual diseases I study in my lab. There's only one here, though, that actually has some direct evidence, Cox postulates, if you like, have been fulfilled for, and that is C. difficile infection. All of the others are associations as such now. I think some of the data for colorectal cancer is now suggesting that that is real. It's another area for another day that I could talk about. But some of these other ones we don't know, but we know that there are some differences in the overall ecosystems of individuals that suffer with these diseases compared to the healthy population. It's actually much harder to look at that than you'd think, because everyone has a different microbiome. So actually looking at these patterns is actually not as easy as you think. So what do we do in the lab? Well, we like to think, first of all, we like to remember that the human gut microbiota is a complex microbial ecosystem. And so if you want to study it in the lab, then its study and its function and its behavior is best studied as a whole. And so how do we do that? Well, it turns out that the human colon is actually a pretty sophisticated bioreactor. And you can actually mimic what's going on in the colon using a bioreactor that's on the bench, okay? And so this bioreactor looks nothing like a colon, but you can create it so that its conditions in the inside of the vessel there are very much mimicking those of the human colon. And at the moment, we're just looking at the distal human colon. Now, of course, it's not a perfect model. I don't think any model is. We're certainly not modeling the mucosa here. We're just trying to provide a situation whereby the microbes are able to function and live within that ecosystem. And then we can start looking at what they're doing in the lab. And so what you have here is the vessel in the middle is kind of the business part of the ecosystem. It's heated, surrounded by a little jacket there of water, which keeps it at 37. That's a really easy thing that we can do. We know our body temperature. We have a pH probe in there. That's just, we keep pH at around 6.8, which is the average pH of the distal colon. We keep the thing stirring very, very gently, just to mimic some turnover of peristalsis and movement of material, and to keep everything stirred up so that there's even distribution of nutrients. We bubble nitrogen gas through it because it's a very anaerobic ecosystem and we want to keep the oxygen out. And actually that's how we make it into a chemostat as well. because the pressure that we are applying there allows us to pump growth medium in and keep it at a level so that the spent medium coming out is always at the same rate. The medium that we use is extremely complicated and takes a little bit of time to make. But basically what it is it's a kind of a mixture of all the things that you might find from the average human diet. We're actually mimicking a vegetarian diet for reasons I can get into later. But what we've done is we've tried to sort of look at what will be left over after your small intestine has pre-digested most of those contents. And so what the food looks like is kind of a disgusting, sloppy mix of mucin and gross things. But the microbes love it. It doesn't smell too hot either, but it smells even worse once the microbes have had a go. So I'm often told, and certainly when I started out in my scientific career, most microbial species, gut microbial species, are unculturable, aren't they? So in fact, one of my mentors when I first started out told me that I shouldn't really do this. It was kind of like a waste of my time, and it was going to be frustrating, and I wasn't going to get anywhere. And then I moved to Guelph, and we got a lot more support there. But it turns out that they are actually culturable. You just have to think about how nature does it. And you think about how we do microbiology traditionally in petri dishes, on plates and things like that, and how removed that is from nature for many of the microbes that we study. Microbes don't like to grow logarithmically, for example. That's not in the gut anyway. That's not their natural way of growing. They are not force-fed nutrients in the way that we tend to feed them rich nutrients in a gut microbiota, in a lab environment. And so we have to really start thinking about how nature grows these things, and that was the ethos for all of this. And so if you actually take this ecosystem, this RoboGut system that we've created, this chemostat system, and you seed it with fresh feces, the system can actually support broad ecosystem growth for several weeks. So what I mean by that, we think, I should just sort of back up a second. What we mean, we've looked at the bacteria, we've also looked at the yeast, we've looked at the archaea, we looked at the virus. Components of all of those, and they're all steady during that period of time. So we can keep these ecosystems going for upwards of 2 months at a time, and without seeing a great degree of ecosystem loss. In fact, it gives us a window of time that we can actually do some really cool experiments. Now we think that this ecosystem reaches steady state, as most chemostats do. When we reach steady state, we do get a drop in diversity from the original fecal sample. And we used to really worry about this because we were like, oh, we're losing some diversity. But if you really think about it, the fecal sample represents everything that you ate for dinner the night before, as well as your resident microbes. And so what we think is going on is that the resident microbes are the ones that are being selected for, because they have the ability to live in that niche. And everything else is kind of being lost to attrition because it doesn't really have, it's a tourist. It wasn't meant to be there in the first place. And this happens fairly rapidly. So, of course, we have no proof of this. It's very difficult to prove that. But certainly the kinds of things that we do lose are the kinds of things that you would find in food or in the environment that probably don't have much of a role in the ecosystem as such. Now, it's a host-free system, as I said, but you can add host components. Mucin is one of them. We don't put mucin in, we get a whole ecosystem collapse. We also have been doing experiments, for example, with secretory IgA, which we can extract from the fecal sample itself, and you can sort of look and see what they do. And any other host components you could think of, if it's available and you can get hold of enough of it, then you could put it into the ecosystem. And you can test the effects of stresses on the ecosystem, and no one gets hurt, which is really great. So from an ethical standpoint, it's, we can put poisons and nasty things. We've done some stuff in the past for someone else with some pesticides and things like that, and no one gets hurt. So we can do these kinds of experiments. We also don't have to ask permission to take samples every few hours. So that's really great. And you can easily measure metabolic output, and that's really key for this this talk, because it's something that I'm going to get to. The nice thing about working with a stainless steel and glass vessel is that none of these metabolites that are being produced by the microbiome are being absorbed by the host. And so they're contained, and we're able to actually sample, so we can actually measure some of the metabolites that would otherwise be quite volatile, and actually see them there in amounts that we can measure. Okay, so at one point we started thinking about, well, could we make an average inoculum? Okay, so how are we going to seed these fecal vessels? And I'm going to show you some data now, which kind of has steered us in the right direction, I think, ever since. So it turns out that everyone's gut microbiota is different, so it's very difficult to ensure that you have experimental relevance if you're just working with a small number of donors. So it's always an issue. So the question is, can you make an average fecal inoculum by taking a whole bunch of donors and pooling their stool together in a kind of a gross mixture, and putting that as to see the robogut. And there have been some studies in the literature that have suggested this is possible, and in fact, that's what they do. But our data actually suggests that it's not recommended. So some work that a very talented PhD student, she's now a postdoc in London, in the UK. This is Julie's work, and she did this a number of years ago when we first started working with the roboguts. She did something that we called affectionately the ultimate fighting chemostat experiment, where we took two different donors. There's nothing particularly special about the donors. They're both two healthy donors, donor 1 and donor 2, and we got their ecosystems from their fecal samples, and we put them up in the robogut, and we grew them to steady state. And then at steady state, we just took both of these things, so half of each of the vessels, and added them together into a third vessel just to see what would happen. And the question was, what would we predict? Would we predict that we would get like an average ecosystem of the two, or would we see dominance of one above the other? Now this work was done some time ago, so actually in those days we weren't using sequencing to look at this, we were actually using HitChip with Willem de Vos's help at Wageningen University. And so what you see here is just a profile from one of the HitChips here. So just for the sake of argument, each of the bands that you see represents a sort of a different species, a different taxon. So you can see there's donor 1 and donor 2, and when you line them up close together, you can see they're quite different profiles. When we mix the two together, so the mixture one now is in the middle of the two, you can kind of see that immediately the donor 1 profile is a more dominant profile, okay? And so this is one day after we mixed the ecosystems together. But by just a few more days later, you can see that the donor 1 profile really took on it was the ultimate fighting champion, if you like, and really outmaneuvered the donor 2 ecosystem. And so for that reason now, when we're working with all of these ecosystems, we will only source our defined communities from one donor, or we will only source a fecal sample from one donor, never from a combined community. There's a lot of things going on that we're just beginning to unravel that are kind of hooking these microbes together within a microbiome using forces that we don't yet understand, but we can't make an average ecosystem because that kind of, that's overcome by some of these forces that we're trying to look at. So that was a nice experiment. I haven't shown the data here, but when we repeated that experiment with defined communities, and I'm going to talk to you about in a minute, so in other words, subset communities from donor 1 and donor 2, we saw the same effect. Now, in the lab, when it turns out that getting poop for experiments can be very hard. I've often wondered why students are so hesitant to give up their poop. Maybe it's because they, you know, a $5 gift voucher for the isn't enough. I don't know, but people are, you know, it's obviously very embarrassing sometimes for students, and I'm not allowed to actually, for ethics rules reasons, I'm not allowed to source the samples from my own students. So we really have to rely on the student body at large, and it can be quite difficult to do. So we got quite frustrated about this in the beginning. Then we also were frustrated because every time we do one of these robogut experiments and we see the species, it takes 30 days to reach steady state, very complicated ecosystems. And every time we do it, even with the same donor, we're going to get a slightly different ecosystem. That's because there's some slight differences between each donation. So we wanted to have some control over that. And we decided that we were going to try and culture some of these so-called unculturable microbes. And in order to make defined ecosystem subsets for each one that we could just pull out the freezer whenever we need to make an ecosystem so that we have a much more reproducible system. And so what we do isn't really rocket science. It's we take a fecal sample and we dilute it and plate it. And that really isn't particularly hard to do. The hard work comes in the fact we're using around 20 different media types. I think it's more than that now, and 2 different atmospheres. And we have just some very talented students, and my technician in particular is extremely good at picking colonies that by the face of it would look exactly the same, but actually are quite different. And we just so that's what we do. And we do this a lot of times. We also, so once we have things on plates and we've streak purified them, we just identify them using standard Sanger sequencing, we curate them and we cryopreserve them. And then we put we can put the ecosystem up in the RoboGut to create that defined ecosystem that we can do again and again. The other thing that we do is at the beginning, when we take the fecal sample, is we actually put that fecal sample into a RoboGut, bring that to steady state, and repeat the whole process of culture as well. And what that does is that allows us to get at some of the microbes which are quite low diversity in store, particularly the ones that you'd normally find in the mucosa. But now remember, we're feeding these RoboGut vessels with mucin, and so we're bringing the numbers of those rare taxa up, and that allows us to capture them with culture, just in this very simple way. I'm very pleased to say that recently the government of Canada took pity on our struggles to try and get as much of this stuff done as we could using these very archaic techniques. And we're now going to get a flow sorter so that we can actually look at things in a little bit more detail and perhaps get at some of the really rare taxa that we've been dreaming about for a long time. So some of these model systems that we create, it turns out, could be therapeutically useful. And so I'm just going to briefly divert just for a few slides to talk about one of these therapeutically useful ecosystems that we've used, and the story of C. difficile infection. C. difficile, for those that don't know, is a gram-positive spore-forming anaerobe that can cause a really nasty form of diarrhea in some patients. For those that don't know how it happens, it's a disease which is a man-made disease from the point of view that it's only really seen as a result of antibiotic use in patients. And so what's happening here is that in a normal person, in a normal colon, you'll have C. difficile. It's either there in very small numbers or it's absent. If you take an antibiotic, and particularly clindamycin or ampicillin or something that's a very broad spectrum antibiotic, then what that does is it reduces the normal gut microbes down to a level, so it's basically killing off everything else. And you can think of C. difficile as a little bit of a weed. It basically grows to high numbers in that situation. When it grows to high numbers, it produces a couple of toxins, endotoxins A and B. They can produce really quite nasty kind of diarrhea and ulceration of the colon, and that can lead to death. Now, it doesn't usually progress that way. Usually what will happen is at the diarrhea stage or the ulceration of the colon stage, a doctor will prescribe vancomycin or metronidazole to bring the numbers of C. diff back down again. The symptoms will go away, you'll stop the therapy, and then hopefully you'll return to normal. But what we're seeing more and more these days is a relapse in around 10 to 20% of patients, sometimes even more, back to C. difficile infection. Because if you think about it, treating a disease which is caused by an antibiotic reducing diversity of the ecosystem with another course of antibiotics doesn't make a lot of sense from an ecological perspective. And so recently, there's been a lot of talk about fecal transplants, which are kind of gross. I'm going to talk about them myself now for a few seconds. Fecal transplants, what are they? They're literally taking poop from a healthy person, putting it in a blender, usually a blender from Walmart, and blending it up with some saline and then instilling it into a patient, into their colon directly, usually by colonoscopy or rectal enema. And in Europe, they've been using nasoduodenal tubes to get it down from the other end, which doesn't bear thinking about. And in Calgary, my colleague Tom Louie's been experimenting with what he calls capsules, which are freeze-dried store preparations that you take orally. And what's really amazing about this, and there have been a couple of clinical trials that have proven this, is that it results in a cure of the patient in over 90% of cases. And some of these cases can be really fulminant infections that could have been going on for a number of months, sometimes even years of suppressive therapy just to keep these patients well. But this treatment it causes very rapid resolution of CDI and only very rare recurrence of disease. The problem is that it's not real. We don't have a Canada Feces Services. The problem is that that fecal transplants are open for abuse, and this is a picture from the internet from a site which teaches you how to do a fecal transplant, like really, and then they put this poop in a blender and put it next to a basket of orchids to somehow sanitize the process. But it's teaching people how to do it, and the thing is, you don't need any special equipment. Everything you need you could buy at Walgreens and take it to your bathroom, find a fit friend and ask who's willing to donate a poop, and you're on your way. And the problem with that is that if you do it without medical supervision, you can imagine there's all sorts of things that can go wrong. And they have gone wrong. I was speaking to my clinical colleagues who've had to undo some of the messes that have happened because of this. And I won't go into that now, but it's pretty scary. Now, there's a clinic in the UK called the Tameout Clinic that sells itself as a medical clinic. It's actually a naturopathic clinic. It serves a purpose for the NHS over there. It does fecal transplants for C. difficile infection. But the thing that I don't like about it is that it's, it's created this kind of myth that they basically keep the stool in quarantine for a few months before they use it. And I'm not actually quite sure, if you think about it, it doesn't make a lot of sense because yes, it may not make the donor sick, but what if it has something in it that could still make the patient sick? That's not going to go away in the 2 months that it's there. Plus the fact that Tameout Clinic, they do this for fee for service, and they've been abusing that a little bit. So they're doing this now for more than C. difficile. Basically, if you pay them enough money, they'll give you a fecal transplant for whatever ails you. And they have done it in the past, I've heard, for short-sightedness. So you can see that there isn't any evidence in the literature anywhere that I can see that short-sightedness is caused by poor gut microbiome. But there you go. So this kind of thing is sort of open for abuse. And then there's also one case study which opened up a whole can of worms about weight gain after fecal microbiota transplantation. So really, just to sum it up, we don't understand the potential negative health implications of fecal transplant right now. And for CDI, the benefits are very clear, right? So you can have this, you always have to have this this benefit versus risk equation when you're an MD and you're trying to make these decisions. But what we don't know about is the long-term effects of the practice. We've never really looked at that, and that could be a problem. So using pure gut-derived microbes as therapy is really the logical next step, and I'm just going to whip through this quickly because we published this some time ago. We created a cleaned up stool, a collection of 33 bacterial strains called Repopulate. We now call it MET1 because journals don't like the name Repopulate. And we took that from one healthy donor. We gave it in a 100 ml dose by a colonoscopy to 2 patients with severe recurrent CDI infection. And both of them recovered within 48 hours, and both have remained C. difficile free to date. So why did we stop? And the reason was because at the time we were doing this as a trial under Health Canada rules as a, excuse me, as a novel probiotic. And it's not a probiotic by definition. And so because it was working, Health Canada asked us to reevaluate this and to make it into a biologic drug. And a biologic drug, to get that through clinical trials, is a much, much harder thing to do than a food supplement like a probiotic. So we created a company, Biota, to do this. And it took 5 years, but we've kind of done it. And the first thing that we asked is, well, which microbial species should be chosen, right? The first one, we met one, it was really what we could grow. And now we really wanted to really sort of think about this in a bit, in a better way. So you look at the weigh scales. You have to consider the needs of the ecosystem, and you have to consider the needs of related to commercialization, because we're a company now. And these things are not necessarily the same kind of things, and sometimes they're at odds with each other. But we try to sort of create a balance between the two. And then, of course, we have to consider the needs of the patient. And the one thing that we heard from them is that they'd much rather take an oral capsule than they would have something go up the other end. And I can appreciate that. Okay, so we started with a donor. We actually found a new donor for this work. We assume that a healthy donor has a healthy gut microbial ecosystem, but we actually don't know, and we still don't know what a healthy gut ecosystem looks like, because everyone has a different gut microbiome, remember. But we can make some estimates based on metagenomic analysis, in other words, what the ecosystem is capable of producing, a little bit like that first slide I showed you with the Human Microbiome Project. And then we can sort of round that up with metabolomic analysis, which is actually looking at the products that the microbiome is actually producing, okay? And so this goes back to this idea that it doesn't so much matter about the taxa that are there, it's what they're doing that's important. So this is some work that was done by a bioinformatics student of mine some time ago now, and I show it just because I still think it's a really good example of the kinds of things that we're doing in the lab right now. So this is using a program called iPath, and we use a different program now, but this is sort of early work. And what this is showing is just basically This is just part of the map of all the different known biochemical functions onto which IPATH will map the gene activities, or the known genes from a metagenome from a given, from what you give it. So what we did is we looked at the 139 HMP samples. So remember, going back to those healthy individuals, those 139 really healthy individuals. And what we did is we colored in black all of the things that we would find in those microbiomes that were common to every single individual in that cohort, 139. And then in green, in shades of green, from dark green to light green, is the degree of sharing amongst the metagenome. So if it's dark green, then the majority of them, of the individuals had it. And if it's light green, just a few did. And you can see that we don't cover everything in this, this map. Part of the reason is because these maps are based on human metabolism, which doesn't help. And so for that reason, it's a fairly incomplete picture. But as I said, this is just a sort of a first look, and it illustrates a point. This is what happens when we start looking at MET1. So this is our repopulate product compared to the 139 HMP samples in aggregate. So the red here is a functional overlap with the HMP aggregate dataset, and the green is in the HMP dataset, but not in MET1. So in other words, those are the things that we missed, that we could probably make a better ecosystem if we included those kinds of microbes. And then in blue, there's only a few little points in blue. Those are in MET1, but not in the HMP aggregate dataset. And that actually allowed us to identify an environmental microbe that we'd isolated from the donor's gut, which actually had no role in the gut, and allowed us to then take it out. And what I want you to appreciate from this sort of mess of a picture, and I don't need you to kind of read it or anything, is just the amount of red that we have just from 33 strains. So I think that's really telling a lot about the functional redundancy within the ecosystem, and the fact that we don't need to create ecosystems that have 200 different species in them in order to create a therapeutic product. The next thing we do is we look at the metabolic output, and for this we look at what we call the liquid gold. So liquid gold, this is the backside of the robogut. Okay, so it's kind of poetic. This is the waste that's coming out of the robogut. It's disgusting, stinky, smelly stuff. But we call it liquid gold because it is by far the thing that I'm requested for aliquots of from labs around the world all the time. It is chock full of the dead microbes, obviously, but also all the things that those microbes have made. And microbes speak to each other using a language of chemicals. And so these chemicals are in this chemical soup. And if we can start to look at chemicals in this chemical soup, we can start eavesdropping on these chemical conversations, if you like. We don't have the Rosetta Stone yet, but the idea is that we can still look at some of the metabolites and look at, see within that, from a healthy person to a diseased person, are there differences in that. And just as an aside, this has actually been the material that has allowed us to culture some of those so-called unculturable microbes on their own as individual microbes. So we took a leaf out of the rumen microbiologist book. Now they use rumen fluid all the time, and they use that as a supplement in their media to help them grow microbes that are otherwise very difficult to culture. All we do here is we filter sterilize this material, this liquid gold, and we add it to our media. And that tricks some of our microbes into culture for the first time, and then it's a nice little trick. So in my lab, so Guelph actually turns out to be Southwest Ontario's Centre for NMR. And when I first joined Guelph, no one used NMR for metabolomics. It's all for structural biology and chemistry. And I needed to put a stop to that because it's this fantastic equipment. So we started working on metabolomics with fecal water. And again, this is one of those moments where people say that that's going to be impossible. There's going to be so much background. How are you going to figure things out? So Sandy Yan in my lab, which is about to defend her PhD, she came in with a chemical engineering background from Waterloo, and she has really made this happen. So what she did in a paper, this is a fairly old paper now, she's done some work since, but again, I just picked this data because it illustrates what she's done rather nicely, is she took an ecosystem, she took 3 different donors, A, B, and C, and measured the metabolites in their fecal water. And we took MET2, which is our second attempt, and that actually came from donor A, and basically measured all the key metabolites using NMR and put them on this heat map that you see here. Now they're separated in sort of space like that just because of the concentration ranges, so that you can really see what's going on. But what I want you to appreciate is that everybody has a different metabolome, which is kind of what you would expect. And also that our MET2 I think at first we were thinking, well, we should really try to match it completely to our donor. And in some cases we have, but in some cases we haven't. And I think we were probably overthinking it a bit, because when we create a defined ecosystem like that, we can't guarantee that we're going to be using up substrates at the same rate that we were using them before. And in some ways, there are some things in there that we don't really want. Like there's p-cresol in our donor. And p-cresol is not a particularly nice metabolite that is produced by some Clostridia. And we didn't really want that in our product. And so it seems like we've got rid of it. So really, this is a useful tool just to make sure that we've got the right kind of concentrations of the right kinds of short chain fatty acids, for example, that we use as measures of ecosystem health. The three major ones being butyrate, propionate, and acetate. And also nothing there that would be, a dangerous thing we don't want to put in people. And so this is shotgun metabolomics. It was pretty hard to start off doing, because we had to learn a lot of how to work with fecal samples and put them into the NMR machine. But we've managed to do it, and I'm glad to say that we're going to be publishing soon a protocol for this. And if anyone is interested in doing this, then please let me know, and we can share the knowledge that we've found and all the pitfalls that we've found along the way as well. Now, for those of you in the audience who might be thinking, well, why are you doing NMR? Why don't you do mass spec? Well, we actually did an experiment a number of years ago now in collaboration with a group in Germany, where we compared the same samples by NMR at Guelph, and he was doing LCMS in Germany. And of course, what you see is that LCMS is far more sensitive. You can pick out far more things with LCMS. You can get proteins, all sorts of things, lots of sensitivity there and greater coverage of metabolites. The thing is, though, when you do mass spec, it's basically a destructive technique. You derive your ions, so you have to break down the product, and then you have to kind of figure out what you've got using a computer. With an NMR, you don't need to do that. So it's a much cheaper way of doing it as well. And it's also inherently quantitative. And so there are some benefits, even though we don't see nearly as many compounds with NMR as we do with mass spec. And just to put that home, when we did some PCA plots of these different samples that we had, so we had 3 different groups of samples. And if you plotted the results in PCA space for LCMS and for NMR, then you got exactly the same kinds of separation. So it may not be that you need to have that amount of sensitivity to see the sort of high level differences between ecosystems. And so we would say that on a high level, if you just want to have a quick look, see, NMR might be the better way to go. And then if you really want to look down, delve in deeper, then you might want to consider LCMS or other mass spec techniques. Okay, so our current thinking behind creating an ecosystem therapeutic is, does the selected ecosystem show stability in the robogut? Is it free from known virulence genes, and that includes antibiotic resistance genes of concern? What does the literature tell us about the gut microbiome for a given disease indication? Is there something missing or overabundant? So we do a lot of literature reviews. And how does the predictive function network look, and how does the actual metabolic output compare? And finally, what can we glean from actual patient data from our trials? And I'm afraid I can't share that because that's confidential information, but certainly that's going to give us the most information and be very exciting. So one of the things that, this is the last thing I'm going to show you, is an experiment that was done by Caitlin Oliphant, and she just defended her PhD and has just started a postdoc in Chicago. What she was interested in is how these ecosystems kind of work together. So in other words, if you're going to put replacement microbes into a person, should they come from a single, or should they come from multiple donors? And it's a little bit like that ultimate fighting chemostat experiment, except in a little bit more detail. So if you think about the first few months of life when you're becoming colonized by a microbiome, your microbes are in a kind of what we call a microbial tug of war. There's a lot of secession going on. There's a lot of infighting and tribal warfare, if you like, before microbes sort of enter a phase where they start to work together, okay? And they gain a sort of a peaceful existence because they're kind of working together to get more benefit. And that's when you end up with your climax community. So we wanted to ask the question, are microbes that come from a single individual co-adapted to each other and therefore cohesive? And if so, does that mean that they're working efficiently together? And so we think that cohesiveness, if it exists, might be critical to ecosystem adaptation. And so we came up with this kind of fun experiment that on paper looked like it would be very simple and actually took 3 years. And what we did is we compared two gut ecosystems. So we had a subset of healthy human-derived microbial community. It's 27 bacterial species. We call that the co-adapted community. And then we had a defined community where all 27 of those CC strains were matched within 97% or greater 16S rRNA sequence identity, usually greater. But each strain, in this case, was sourced from a different healthy or different donor fecal sample. So 27 different individuals contributed to this ecosystem. And we called that Frankenstein's community, because all of our experiments have to have a cool name. And we reasoned that just like Frankenstein, this community might be a bit more lumbering in its ability to cope with what you throw at it. So the first things first, and this is another aside, but something that's quite important, is the reason this experiment took 3 years to do is that when we started out, we thought it was very important to have pure isolates, right? Because you need to know that they're absolutely pure. And it turns out that pure isolates that you get from the human gut may not be pure. So we do a lot of dilution to extinction and streaking to purity to get purity. And it turns out that might not be sufficient to separate species that might have evolved, for example, a close bond with cross-feeding or whatever. And so what we had to do is we had to take each individual isolate and we deep sequenced it with 16S RNA with a V3-4 region for every single strain used for this experiment to get at least 80,000 reads for each. And we looked at the numbers of reads, and only if we reached a certain threshold that just exceeded the error rate of Illumina, we had did some complicated mathematics to figure this out, would we deem that something was pure? For several strains, it turned out that there wasn't purity. We detected these sort of contaminants. We're hesitant to call them contaminants. We prefer to call them friends. And they're at an equivalent rate of around 1 in 10,000 cells. And the important thing to remember here is that these contaminants were real. Once we knew what they were, and we could go looking for them and use selective techniques to get them out, we could isolate them. And so they are there. So we have to be very careful. And what we've found since is that there are, in fact, some strains in strain collections around the world that have come from the human gut, which are also not pure. And so it's something that is a sort of a cautionary tale. We now do this kind of level of sequence analysis on every strain that we're going to use in these kinds of experiments, because it's quite an important thing to remember. Okay, so going back to the Frankenstein's community experiment, what we did is we set our communities up. We fed them either a high fiber diet or a high protein diet, and we allowed them to reach steady state. And then we switched the diet halfway through, and we then did 16S RNA gene profiling and metabolomics. Okay, so in this particular experiment I'm going to show you, we started off with a high fiber diet, and then we switched it to high protein. This is what we saw, right? So the co-adapted community relative abundance doesn't change much when the diet changes, all right? So this is just what we're seeing in abundance profile. These are normalized. So you can see just at the phylum level. But look what happens to the Frankenstein community. We don't see quite as much stability. And in fact, the thing that is kind of taking over there is the proteobacteria. I mean, you might expect that because proteobacteria are a Jacksonville trait. They can kind of cope with anything that life throws at them. And so they kind of, they come up in number. When you look at the family level, you can see, just in a slightly different way, you can see the co-adapted strains are very kind of level. Nothing's really going on with them. But again, the Frankenstein community, you can see fairly major changes in terms of abundance as the experiment goes on. So then the left again is in the high fiber diet, and then the right is when they're switched to a high protein diet. And then at the species level, what we did is we looked very fine detail, and compared to the cohesive community, the Frankenstein community lost Coprococcus comus and the Eubacterium rectale. These are major specialist degraders of dietary fibers that we find in the gut that cross-feed with each other as well. In general, the Frankenstein community was impaired in its ability to degrade fiber. And instead, we saw an increase in the products of stickland fermentation. This is specialized amino acid fermentation. It's very sort of what we call selfish fermentation, because it takes place with just within one species and allows them to gain more energy. It suggests that polysaccharide utilization by Firmicutes might be dependent on species efficiently working together. And if you don't get cooperation, there might be a switch to more putrefactive or bad pathways, not necessarily good. So we're probably right to source each of our therapeutic communities from individual donors. But do remember, though, this is a model, a very sort of an early model too. And we did only have 27 species. So, you know, we don't know when we increase the amount of diversity in there, whether we'd see a similar kind of thing. But it does point to this sort of cohesiveness and co-adaptation. So just to finish off, moving towards the future, how should we approach the development of novel mets? So I've kind of visualized it like this. So we need to look at the gut microbial engine. Okay, so this is actually the engine of a Mini, which is my first car. And I like to think of the microbiome as a sort of having a blueprint, right? Your microbiome has a blueprint, a little bit like an engine of how it works. And when your engine isn't working well, you take it to the shop and what do? They measure the emissions to see how it's working. And then they, based on that, use their complicated computer algorithms to tell you which part is faulty, and then you replace that faulty part and everything should work well again. And we can kind of conceptualize that also in the same way for the microbiome. So now we have a metagenome, that's our metabolic engine blueprint, if you like, and the metabolome, which is the metabolome. Metabolome is a new word in the lexicon now. So the metabolome is like the emissions. That's what the microbiome is actually producing. And so in the future, I think that we'll start looking at the metabolome as the first clues that something is not right with a given ecosystem. And then we'll start to replace missing functions by adding different microbes or different metabolites to get the engine running optimally again. And I think that's fundamentally different from what we're doing now, which is creating ecosystems and throwing them in there and just seeing what sticks. I think in the future we can do this in a much more directed way. We just need to understand a little bit more about how these microbes work together in ecosystems in order to do that, and that's what we're doing. So we can start to understand the importance of microbial cooperation in this process, as I just showed you, and all sorts of other things that we're doing using these kinds of in vitro models, which I think is very important. So in summary, redundancy in an ecosystem is really important, and it fosters ecosystem stability. Development of microbial ecosystem therapeutics, as we call it, needs to be based on rational design, and in turn based on ecological principles, and we're not there yet, but we're starting to make the first steps towards that. And studying these principles is possible using Bioreactor model platform and measuring, for example, ecosystem composition and function. And we've determined that members of the gut microbiome might be co-adapted to each other, and this should be respected as new ecosystems are developed. And with that, I'm just going to acknowledge all the people in my lab that do all of the work. And this was at a recent rare get-together where I think almost everyone is in that picture, which is really kind of cool. And then all of the people that fund me, and thank you to you all for your attention. And I'll answer the questions.
So great talk. And what I was curious about was your interpretation of the co-adaptation you were observing as cooperation.
Yeah.
Because I remember reading a review from Kevin Foster where the stability of the gut microbiome seems to depend on a lot of negative feedback and not a lot of negative interactions that were occurring between members. So I was wondering if you're able to comment on your results in that framework or vice versa.
Yeah, so I had this question at another meeting, and I didn't really have an answer for it then. I think that there's probably many different forces working in this situation. I remember that particular model ecosystem was a very pared down model. So I imagine that when you've got more diversity in that some of those, the forces that we're seeing will become, it's difficult to know the size of which force relative to each other, right? And so maybe what we're seeing is something that's a little bit more blown up because we're looking at a very small ecosystem compared to what it would actually be in real life. And so with more diversity, then those adaptations might become less of an issue. So I think that's probably the reason there. I think we're really interested in looking at these kind of pared down ecosystems from the point of view of ecosystem therapeutics, because we can't make an ecosystem that's 200 species, right? And be able to make a profit in terms of, or even be able to do it, physically be able to do it commercially. So we're trying to just use microbes that have a better chance of working together as a in synergy within an ecosystem than just ones that we pick off a shelf and put together. So I don't think that answers your question nearly as well as I could or should, but I don't really know is the answer, but I suspect it's because it's just about the amount of diversity that's there. Yeah.
I think I told you earlier that the last time I saw you, I'll have to do it again. I wanted to ask you about the thinking public transients. You're talking about microbes that come and then sort of disappear. Maybe those aren't important. We had another speaker who talked about digging ant colonies out of the ground, and when they got to the lab, they saw this sort of convergence. And similarly, we're thinking of those organisms as transients. And we're wondering, are organisms like that, are they important still? Like if you think about metapopulations as sources and sinks, things that are existing in the sink, they still perform important functional roles, but they're just not self-sustaining in that way. So I'm curious, like if you think there are things that are transient, but they just require sort of a constant source of source.
Yeah, that's a really good question, and I would say that we're very dismissive of the tourists, but because that sort of fits our model, right? But I think you're right. They absolutely could be doing something, and I think the best example of that would be like a probiotic, actually, which is your archetypal tourist organism. They don't colonize, but some of them have effects and clinically recognized effects. So how do they do that? So as they're passing through, they might have some metabolic effects or something might be happening, and so that's a good example. I think there's all sorts of work out there about the effect of the environmental microbiome on the human microbiome, and owning a dog that licks your face often is really good for your microbiome. I don't know in the context of that whether those microbes would be tourist microbes or we're talking about establishments or whatever, but I imagine it's very similar, and that perhaps during that period of time those microbes are producing some effect within that ecosystem. And it's very much like tourists within a city. You go into a tourist and they're not doing nothing, they're contributing to the economy, so they're actually doing something while they're there. So yes, I am dismissive of them, but only from the point of view that we're kind of moving more towards defined communities now just for our own sanity. But yeah, you're quite right.
I have a question about your NMR technique.
Yeah.
Would you mind going to it's sort of like a Venn diagram with
Yeah.
your PCA? Probably halfway through. I think that's really powerful.
Yeah.
There you go. So is this implying that the NMR detected different compounds
Yes.
that were not
Detected, yes.
by LCMS?
Yes. So the compounds that that NMR is particularly suited to are small molecules, and some of those small molecules are not detectable by LCMS.
I see. Okay.
Yeah. So you can measure the short chain fatty acids, for example, using both techniques. Those are within the 22 there, but there are some which are definitely not discoverable by mass spec. Yeah.
It all seems like the therapeutic goal is to replace the microbiome with a healthy one. Are you or anybody working on sort of directed modification by species to get back to the
What do you mean by fewer species? Like,
it seems like you said you couldn't create an extra way to get back to 200.
right. Yes, right, yeah.
Could you take the disease state at 0 and get back to that 200? Or could you
I see. Yeah, I see what you. Yeah, so it's kind of what we're trying to do with the ecosystem therapeutics that we're doing right now. We're just not doing it in a very predictive way because there's a lot of pressures on even getting a product. It's a first-in-class biologic, so there's lots of things we've got to do to get that. I think in the future, eventually, as I said, we'll be looking at what people have, and it will be on a much more individual basis, right? That's the key here. It's got to be personalized medicine. And we'll be looking at what people have and try to figure out, in that situation, you are missing this function and this function and this function, and you probably need this microbiome. Whether or not we can just replace it and keep it there for good, I think is the key question there. And everyone's trying to answer that question. In my opinion and our experience so far, that is not possible. And I think that it's probably more of a like a Band-Aid effect. And you would need to keep providing the microbes. So we don't understand the forces that are involved in that. It's clearly more than just ecology, because if it was ecology, they would fit the niche and should be all fine. but there's probably some host effects there. We're looking right now at the effect of secretory IgA on the ecosystem and the effect of the bacteriophages, which I think are two of the other driving forces that I didn't have time to talk about. But I think, one of the other questions actually that this question raises is that we don't know long term. So if we keep giving a product to a person, whether or not they would then adapt to that ecosystem, and how long would we have to do that for in order for those microbes to become adapted within that ecosystem? That's an open question and one that's absolutely fascinating, but I think we have to kind of get there in baby steps. Yeah.
So I was wondering, you developed a couple of these different therapeutic communities from different donors. And when you were showing different metabolic functions, you were saying that you were missing some. Is it consistent which ones you're missing?
Okay.
With different communities?
Yeah, and I probably misspoke a bit because you remember we were running these in the RoboGut under one set of conditions. So whether or not something is missing or not is... Yes, that's right. So if we really wanted to examine that in any detail, we'd have to work with lots of different diets and lots of different ways. We started to look at the fecal water rather than the RoboGut water, if you like, from the donor. The problem there is that you get a massive amount of signal, which is from metabolites, which are actually from the human side of the person. So it's very difficult to kind of see through the murk, if you like, to see what's there. So I'm not sure whether if they're missing, they're truly not there, or we're just not, they're not being expressed at that time under those conditions. Does that make sense? Does that answer your question? Probably not.
Yes, well, but then do you also, so that's from metabolomics.
Yes.
But do you also then look metagenomic for capabilities?
Oh, I see.
You get
I see.
Capabilities.
Yeah, so that's so when we're looking at this kind of thing, the difficulties of those maps, those crazy maps I showed you, the difficulty is that when you sequence the metagenome, there is so much dark matter in there that we have no idea what it's doing. We don't know what the genes are. I mean, we can predict their genes, that's about all we can do, and we don't know what they do metabolically. And so what we're looking at when we look at that kind of predictive modeling is really the tip of the iceberg. And I suspect we're going to see an awful lot of other things going on. There's probably many alternative pathways to many metabolites as well that we're not seeing. So we did that as a way to visualize how we want to do things in the future when things improve. But I don't think, it didn't really pan out that as well as we hoped just for using iPath, for example. So we're using some other tools right now, which are slightly better. But I think the key problem here is that we just know so little about the genomes of these things. It's kind of why we went to what are they actually producing, because that is real. Right? Thank you. Oh, I don't know how to check my disk.
All right, okay.
No.
Got it wrong, but
Okay.
great talk.
Thank you. Good
Very
to see
nice.
you. Thanks for tuning in.
Yep. Yeah, it was nice
Okay.
talking to you.
Thank you.
Yeah, thank you. Okay, I appreciate that. Yeah, he was saying like I immediately
I don't want to run around all your stuff. Maybe we should talk over.
thank you, but I will do it.
Of course. We might need to
Yeah. I appreciate you.
It's kind of expensive.
And is that you purchase it?
Pardon?
Is that the small stream?
I think so, but yeah,
Oh, yeah, yeah. I'll be here. Thanks. Thank you.